So we recently published our data on evaluating ctDNA compared to tissue in patients with a proven diagnosis of advanced non-small cell lung cancer who were having standard treatment with tissue genotyping, and at the same time we implemented ctDNA analysis done after the confirmation of pathologically verified advanced non-small cell lung cancer. We published our findings in the European Journal of Cancer, and in this cohort of just over 200 patients we were able to identify that ctDNA brought a lot of advantages to the treatment paradigm. It identified more tier 1 actionable variants compared to tissue alone. It did have some limitations because it did miss some variants that tissue picked up, but similarly, tissue missed some variants that ctDNA picked up. Both should be considered complementary, but importantly, ctDNA using a commercial platform gave a faster readout of genotype than awaiting for tissue genomics. That allowed patients to start their first-line therapy much earlier than waiting for tissue NGS. So in our data set, our opinion is that ctDNA in parallel to tissue is complementary, and one could and should consider a plasma ctDNA-NGS based strategy upfront, potentially supported by tissue in those that need it for routine therapeutic decision making in advanced non-small cell lung cancer. And I'm delighted to see that the recent publication from the ESMO Precision Medicine Working Group looking at the clinical utility and best practice in ctDNA implementation has also concluded the same findings for non-small cell lung cancer.